Good
Good
morning.
morning. So Ahmad and I will present some result of 4 projects, I think, on the optimization of treatment by AI. We start working together 3 years ago.
3 years now, yeah.
And we will show you some cases and some results. So why glioblastoma? So glioblastoma is the most frequent and aggressive brain tumor in adults, and the incidence is increasing. And despite surgery, radiotherapy, and chemotherapy, the patient will relapse very rapidly in a few months, and leading to a median overall survival around 16 months. So very short, unfortunately, because of this recurrence. And now we a little bit increase the overall survival of this patient, adding some electric fields. So you can see here what it looks like, glioblastoma and MRI. And the first project aimed to study by AI very important prognostic factors, the methylation of a gene, MGMT, which is associated with a better response to radio and chemotherapy, and a little bit increase of overall survival for this subgroup of patients. So because this study of the MGMT methylation is done now by biopsies, and that the biopsies are not necessarily representative of the anti-tumor and can vary at relapse, the first question that we asked with Ahmad was, can AI apply to MRI determine precise MGMT methylation status? So, Ahmad.
Thank you. Indeed, this was, at first we thought this was an obvious application for AI algorithms on MRI data. So we started looking at what we could find in literature, and there were many publications that said, okay, this can be done easily, and with impressive results. We wanted to verify that using data from Oncopol, from Elizabeth. And we were very surprised to see that, in fact, it did not work at all. And we tried to investigate why we had these strange results. So the first thing we tried was simply a binary classifier to see if using public data and or private data, we were able to try and reproduce these things, but it was not possible. And then we tried the hypothesis that maybe it depends on the degree of methylation. Maybe this was something that was only visible with higher degree of methylation, but also this was not possible. As you can see on the different precision recall curves here, we had similar results, even with higher degrees and lower degrees of methylation. So we published a paper about this to explain why we didn't see this difference. And also we tried to understand how other researchers managed to come up with impressive results. And the thing that was most common was data leakage when they were using 2D slices from MRI. So they sliced the brain in 3D. They used 2D images in the training set, and and other 2D slices from the same patient in the test set. But these were highly correlated. So this kind of data leakage, unfortunately, was very common amongst the publications that had very interesting results. And it explains the contrast that we saw in this process.
Okay, so next, thank you, Ahmad. So the next project that we developed together was to discriminate by MRI and using AI a subgroup of glioblastoma. Indeed, as you can see here, that's a standard glioblastoma diagnosis on an MRI with a contrast enhancement and an invasion part with what we call the flare sequence. But in fact, there is a subgroup of glioblastoma patient with a bad prognosis, which looks exactly like another tumor, which is low grade with a totally different prognosis. You see that it's not possible for us and for a neuroradiologist to discriminate this tumor from this tumor, but they are not at all the same. This one is a glioblastoma with a very bad prognosis, and this one is a low-grade glioma with a good prognosis. So it's very important to be able to discriminate it. And the second question was, can AI apply to MRI see some things that the professional eye cannot? So
So in essence, Elizabeth wanted me to try to do what she could not do.
Exactly.
And this time,
And
and this time the answer was yes. So we were very happy to see that our, well, using convolutional 3D networks like ResNet 3D, we were able to successfully distinguish these two types of glioma. And this is in a publication preparation with our PhD student and your intern.
Yeah.
Right. And I will detail a bit how we train these models in the upcoming applications, so I won't repeat myself again.
Okay. So the third project is the study of a prediction of relapse for all the patients with glioblastoma. Indeed, as I told you at the beginning of my talk, all the patients will relapse very rapidly, but the relapse is different according to the patient. You can see here the primary tumor. Some patients will relapse locally in the treatment fields of radiotherapy, but locally, it's the same place. And some will relapse in the brain always, but a distant relapse. And we think that these different patterns of relapse are linked to different biological pathways and genes involved in this relapse. And as you can see here, these biological pathways could be linked to the presence of glioblastoma stem cells. These stem cells are the roots of the tumor, and they have a high plasticity after treatment to adapt and leading to kind of recurrence. You can see here the heterogeneity of a glioblastoma on an MRI with multimodal imaging. That's a classical anatomical contrast imaging. Here you see the heterogeneity of perfusion. You see here some spots of hypervascularization and some are not. And interestingly, we study a lot in my department the metabolism of the tumor, and we are able in 3D, but using MRI spectroscopy, to map some metabolites involved in aggressivity. Several years ago, you can see here an hypermetabolic area which doesn't correspond to the contrast enhancement, so you see the heterogeneity. Several years ago, we showed clearly that this area was predictive of the site of relapse after treatment. So my first question was, okay, if it predicts the sign of relapse, maybe it could be enriched in glioblastoma stem cells. And for that, I developed a clinical trial with a surgeon. And what we did, it's during the resection, the surgeon takes some biopsies of the different parts of the tumor according to the metabolic area. So in the infiltrative zone, metabolic or not metabolic part of the tumor, and the same thing in the contrast area. All the biopsies were sent to my lab. We perform on the biopsy RNA sequencing, so we get a lot of genes, expression and mutation. We perform also an extraction of glioblastoma stem cells coming from each part of the tumor. And each stem cells, we perform also an RNA-seq on it. All stem cells were implanted in brain mice to study the aggressivity, the aggressiveness, invasion, the vascularization. Of course, we got the mice survival, but also the patients were treated after surgery by radiochemotherapy, followed every 2 months by multimodal imaging until relapse, and we got, of course, the pattern of relapse of the patient. So we have a lot of data. We first showed that our hypothesis was good, right? We clearly showed that in the metabolic area, this area is enriched in glioblastoma stem cells with a high expression of a lot of genes involved in aggressivity, like DNA repair, stemness, invasion, angiogenesis, and cell cycle. So we just published it, and we are very proud in Science Advance 2 weeks ago. And then what we want to do, what we develop with AMAD now, is to be able to predict at the beginning of the diagnosis using imaging and the slice of the tumor to predict the pattern of relapse and the biological pathways involved in this pattern of relapse, so that one day we will be able to say, okay, for this patient, he will relapse with such pathways, and we will use then a new therapeutic agent associated with treatment against this pathway. That's our new goal. And Ahmad.
And yes, this was very challenging. This was here we really see the power of AI in handling this this type of data, because here we have to deal with multimodality. It's not just imaging. So at first we needed to encode RNA-seq data. So that you have a bit of context, at IRT we mainly deal with aerospace applications and spatial applications and also defense. So when I saw RNA-seq data, that was something new to me. But still, these were vectors, so we treated them as we would with text. So we trained a token, a self-attention model, like a bit like masked language modeling, using contrastive learning. So we were able to predict parts of RNA from other parts of RNA, to make it simple, like as if they were words in a sentence. So that worked, because what we tried to do is to look at their representations in 2D that came out of our model. Here you can see the 2D distribution of the output of our model for different types of RNA-seq data. So some come from the brain in blue. They are mainly here. Like you see, it looks like a bit like France. So it's like where there's Corsica. And the other is, well, the other types of tissues are a bit distributed in the other parts. And we wanted to verify that the data that Elizabeth gave us match what we could find in public data in the TCGA Cancer Genome Atlas. And this is what we got. So the data from Elizabeth also matches brain data. It's in Corsica. So yeah, that proved that our technique for encoding RNA-seq data worked. Okay. And then we turned to the mice data. So as Elizabeth explained, we had mice brain slices. So you can see that in the lower left of the screen. In red, you have cancer-invaded cells in brown. So these are the red squares. And the green squares are normal parts of the brain. So we took these patches. We put them in a multimodal encoder that we use to match images against captions of images. And here our images are the mice brain, and the text is the RNA data. So we use techniques used in text language modeling. Here it's ALBEF for you who knows, for those of you who know that. It's aligned before fuse. So we aligned the modalities using contrastive learning, and then we fused them in a cross-attention model. And then we wanted to see if these matches for we were able to match mice data with RNA-seq data for a same patient and for same lineage of stem cells. And these are the results that we got. Well, they're here. So we were able to match successfully RNA-seq data and mice and microscope imagery from mice with 97% accuracy. So matching was perfect. And then we wanted to see how our model managed that. We wanted to see, using explainability techniques, how this, what led to this perfect matching. And this is interesting because this explains then, this gives us an insight into the pathways that are involved in aggressivity when considering patients with lower survival rates. And this is what we got. So this has not been published yet. Please keep it to yourselves. We were able to identify new genes and new pathways that explain this matching. And some of the genes that were identified have already been mentioned in literature, but there are also new ones.
Okay, so the next project, as I told you, every patient are going to relapse after radiotherapy. And in some case, one of the main weapon in case of recurrence is to re-irradiate the patient with another technique. So we can, as you can see, that's the first planification of radiotherapy. Then at relapse, it's exactly in the same area. We are able to add 3 fractions of very high dose of radiotherapy, 3 fractions of 8 or 9 grays. And this kind of fractionation have been shown in preclinic to boost the immunity, to induce an immune cell death. So that's why we decide to develop a clinical trial associating this kind of re-irradiation with checkpoint inhibitor, so immunotherapy. First, we perform a phase 1 trial on 6 patients to study the tolerance of the treatment. So we showed that it was very well tolerated. And we got, as you can see here, for recurrent glioblastoma, starting the recurrence, very encouraging results. But what we wanted to see, of course, is to do it on a bigger population. So we perform and finish the phase 2 randomized trial, randomizing radiation with radiation plus immunotherapy, durvalumab. It's an anti-PD-L1 treatment. And because all the patients are not going to respond to this association the same way, of course, we are all different. So we wanted to develop ancillary studies. It's a national program which has been granted by the ARC Foundation. And for all the patients, the 100 patients, all were followed, of course, every 2 months before and during treatment until relapse by multimodal imaging. We got also a lot of blood samples in the patient before treatment, then every 2 months until relapse. And also we analyzed the tumor of the patient. So on the blood samples, we looked at a lot of data, cytokines, exosomes. Exosomes are small vesicles for communication between cells. And in these vesicles, you have a lot of genes. And we perform RNA sequencing on the exosome of the patient. So a lot of data. And of course, our goal is to predict at the beginning of the treatment the efficacy of radiotherapy and immunotherapy, so to choose the patient who will benefit from this treatment.
So yeah, this actually was the first study that we conducted together. And here is the core of the techniques that we used at almost every step of our work together. Because, as you already know, patient data is not very common. It's very valuable, and it's hard to get. There is public data, but we need to leverage it as much as possible without considering all the labels. Because if we start doing supervised learning at the beginning, we lose every means to generalize easily. So we apply contrastive learning using multimodal data at every step. We do this at first using MRI data. So what we do is we match some of the modalities of one patient against the modality of the same patient. So this is a positive match. But we also contrast it with the modalities of other patients. So this is a negative match. And using this type of triplet matching, we are able to learn in an unsupervised or weakly supervised way meaningful representations of MRI data. And then at the very end, this is where we focus on the labeled data. So we match these representations, that were obtained in an unsupervised way, using the labels, which in this case is the survival or the progression-free survival interval. So this is the key recipe for dealing with multimodal MRI. We also take into account clinical data. And in this case, cytokine data and data from from blood samples like the lymphocyte ratio and neutrophil ratio, neutrophil to lymphocyte ratio. Thank you. The other way around. And we then clustered this data. We did that on the phase 1 data, but we have pre-trained our model using public data. So we didn't only have 6 patients. Actually, training was done over a lot of public patients just for the MRI part. And then adding cytokine data, we were able to clusterize patients without any consideration for their survival rates or relapse rates. And we were very surprised to see that the clustering that we obtained matched what we saw in either overall survival or also progression-free survival. So what you see on the left is the overall survival, and the patients 2 and 5 that had the highest overall survival were put in the same cluster. The reason why you see multiple data points for the same patient is because for a single patient you have multiple visits and multiple MRIs. And what's interesting to see, because since we use metric learning, we are able to measure how patients are different from one another. And this is what we saw, what we see on the right. So for a same patient, for example, patient 4 here, you can see from the first visit to the second visit, things evolve. So they begin with less aggressive disease to something more aggressive. And the same can be seen for other patients. Maybe you want to add something else?
No. It's very what is very interesting. Of course, Ahmad didn't know the overall survival of the patient, and for this patient, the patient 2, in fact, we thought at the beginning that after radiotherapy and immunotherapy, in clinic you can get what we call pseudo progression. So on an MRI, you see an increase of the lesion that you treated, and we thought that. Probably it was a progression, so we stopped the treatment. So the patient was then treated by another treatment, and in fact it was probably a pseudo progression because the patient had a longest survival of the trial, and he died from COVID and not from his recurrent glioblastoma. So what is very interesting is that Ahmad with MRI and cytokines was able to tell me that, you know, this patient is like the other one, is different from the other one. Yes, he was different. He survived longer. So it's a very powerful tool. And I'm pretty sure that, of course, now we are going to confirm all this data with a PhD of Luca on the 100 of patients already included in this trial.
Exactly. So we were very surprised to see that. Actually, we thought it was a mistake at first, and then, yeah, we were...
No, it was not a mistake.
And the future.
So, yeah, the future. So, as I told you at the beginning of my talk, now the third treatment that we give to our patient, it's a treatment by electric fields that after radiochemotherapy. And interestingly, when we know the mechanism of electric fields, and also of radiotherapy, they have multiple synergistic effects, and that we study also in my lab. And clearly, we have shown that association of radiotherapy and electric fields led to an increased efficacy on glioblastoma stem cells. So combining it, that has been also studied in an international clinical trial also where we participate a lot, is very interesting. And interestingly, what we have shown in the lab also is that radiotherapy and electric fields have common radio-resistance and TT field resistance pathways, so common biological pathway with new targets, etc., new mechanisms that we patented with the company. And now, of course, that's in the lab. Now we want to see the real life and to validate our pathway of sensitivity or resistance in the patient treated in my department by radiotherapy and then with TT fields. And again, we designed a new clinical trial, ExoField, which will start beginning of next year. And as we usually do, we will perform multimodal imaging, but also blood samples before and during treatment, during radiotherapy and during TT fields. And by using AI with Ahmad, we will, I hope so, validate what we found in the lab, but also probably bring to light also new targets of sensitivity or resistance to TT fields, then to, in another step, optimize the treatment by adding targeted drugs against this pathway. That's the future. So thank you for your attention. I wanted to thank all my patients and the family, my research lab, my department of radiotherapy, all the neuro-oncology committee with neurosurgeon also involved in research, medical oncologist, everybody, of course, all the team and Ahmad. It's a real pleasure to work together. So,
indeed, it is.
And I would like to thank, because we need money to develop all this research. So all the foundation who funded our research. So thank you for your attention.